Hello, everyone. Welcome back to Meds.Concepts. Today we are going to study one of the most important and conceptually rich topics of molecular biology, genetic recombination. Recombination is a process in which DNA molecules exchange genetic information, producing new genetic combinations. This process is essential for genome stability, DNA repair, chromosome segregation, evolution, and viral integration into host genomes. In this lecture we will mainly focus on site-specific recombination, serine and tyrosine recombinases, lambda phage integration and excision, Cre-loxP system, and double-strand break repair pathways including NHEJ and HDR. Introduction to genetic recombination. Genetic recombination refers to the exchange of DNA segments between DNA molecules resulting in new combinations of genetic material. Recombination is biologically very important because it generates genetic diversity, repairs damaged DNA, helps proper chromosome segregation, and contributes to genome evolution. There are three major types of recombination: homologous recombination, site-specific recombination, transposition recombination. Homologous recombination requires extensive sequence homology. Site-specific recombination occurs only at short specific sequences recognized by recombinases. Transposition involves movement of mobile genetic elements. Depending on the mechanism, recombination may produce integration, inversion, deletion, or translocation. Site-specific recombination. Site-specific recombination occurs at short-defined DNA sequences called recombination sites. Unlike homologous recombination, it does not require long homologous regions. Specialized enzymes called recombinases recognize these sites and catalyze DNA rearrangement. The process is highly precise and sequence-specific. Major outcomes include integration of DNA, excision, inversion, and translocation. Site-specific recombination is widely used by bacteriophages, plasmids, transposons, and modern genome engineering systems. Important enzymes involved poses nuclease activity for DNA cleavage, and ligase activity for strand joining. This mechanism produces highly precise DNA rearrangements. Classes of site-specific recombinases. Site-specific recombinases are divided into two major classes: serine recombinases and tyrosine recombinases. Serine recombinases use an active site serine residue during catalysis. These enzymes cleave all 4 DNA strands simultaneously. Tyrosine recombinases use an active site tyrosine residue. They cleave and exchange DNA strands sequentially. Both enzyme classes cut DNA, exchange strands, and relegate DNA. However, their strand exchange mechanisms are fundamentally different. Serine recombinases mechanism. Serine recombinases catalyze recombination through a rotational mechanism. First, the recombinase binds specific recombination sites. Then, all 4 DNA strands are cleaved simultaneously. A covalent serine DNA intermediate forms. Next, the DNA subunits rotate approximately 180 degrees relative to one another. Finally, DNA strands are relegated, completing recombination. This mechanism is very rapid and efficient for DNA inversion reactions. Examples include HIN invertase and GIN invertase. A very important point is that serine recombinases do not form Holliday junction intermediates. Tyrosine recombinases mechanism. Tyrosine recombinases operate differently from serine recombinases. These enzymes cleave one DNA strand at a time. The active site tyrosine attacks the DNA backbone and forms a covalent phosphotyrosine intermediate. After the first strand exchange, a Holliday junction intermediate is formed. Subsequently, the second pair of strands is cleaved and exchanged. Finally, the Holliday junction is resolved, producing recombinant DNA molecules. Examples include lambda integrase, Cre-recombinase, and XCR recombinase. An important feature is that this process usually does not require external ATP. Lambda phage integration. Bacteriophage lambda integrates into the bacterial chromosome using site-specific recombination. The key enzyme involved is lambda integrase, which is a tyrosine recombinase. Two attachment sites participate: attP present on phage DNA and attB present on bacterial DNA. Recombination between attP and attB generates attL and attR sites. Integration also requires a host protein called IHF or integration host factor. IHF binds DNA and introduces bends that facilitate recombination complex formation. Thus, lambda integration depends on both integrase and accessory DNA bending proteins. Lambda phage excision and exciase protein. When lambda phage enters the lytic cycle, the prophage must excise from the bacterial chromosome. This process requires lambda integrase, IHF, and another phage protein called exciase. Exciase stands for excisionase. Exciase binds specific sequences in attR and bends DNA. Its functions include stimulating excision and inhibiting integration. Thus, exciase acts as a directional regulator controlling recombination outcome. Importantly, exciase protein is synthesized only during lytic induction. Structure of ATT sites. The attachment sites involved in lambda recombination possess defined structures. attB is the bacterial attachment site and is relatively small, approximately 21 base pairs long. attP is the phage attachment site and is much larger, around 250 base pairs. Both sites contain a common core sequence called O. Their organization is attB equals BOB prime and attP equals POP prime. Recombination occurs precisely within the shared core sequence. Correct alignment of the core region ensures highly accurate recombination. Cre-loxP recombination system. One of the most important tools in modern molecular biology is the Cre-loxP recombination system. Cre-recombinase is a tyrosine recombinase derived from bacteriophage P1. It recognizes LoxP sites, which are 34 base pair DNA sequences containing 2 inverted repeats and a central spacer region. The orientation of LoxP sites determines the recombination outcome. If LoxP sites are in the same orientation, the intervening DNA is excised. If they are in opposite orientation, the DNA segment undergoes inversion. If LoxP sites are located on different chromosomes, translocation can occur. Cre-loxP technology is extensively used for conditional knockout generation, tissue-specific gene deletion, and genome engineering. Double-strand break repair. Double-strand breaks or DSBs are among the most dangerous forms of DNA damage. DSBs may arise due to ionizing radiation, oxidative stress, replication errors, or chemical mutagens. If unrepaired, double-strand breaks can cause chromosome fragmentation and cell death. Cells poses to major repair pathways. Non-homologous end joining or NHEJ. Homology-directed repair or HDR. The choice of pathway depends strongly on the cell cycle stage. Non-homologous end joining, NHEJ. NHEJ is an error-prone repair mechanism because it joins broken DNA ends without using a homologous template. This pathway is active throughout the cell cycle but predominates in G0 and G1 phases. The first proteins recognizing broken DNA ends are Cu70 and Cu80 heterodimer. These proteins recruit and activate DNA PKCS. Next, Artemis nuclease trims damaged DNA ends. Finally, DNA ligase 4 together with XRCC4 joins the DNA ends. Because no template is used, small insertions or deletions frequently occur. Therefore, NHEJ is considered an emergency repair pathway. Homology-directed repair, HDR. HDR is an accurate error-free repair mechanism. Unlike NHEJ, HDR requires a homologous DNA template. This pathway mainly operates during S and G2 phases because sister chromatids are available. The major steps include end resection strand invasion DNA synthesis, Holliday junction formation resolution. A key protein in strand invasion is RAD51. BRCA1 and BRCA2 also play essential roles in homologous recombination repair. HDR maintains genome stability with very high fidelity. Comparison between NHEJ and HDR. Now let us compare NHEJ and HDR. NHEJ does not require a homologous template, is fast, and active mainly in G0-G1 phase. However, it is error-prone and may introduce insertions or deletions. HDR requires homologous DNA, is slower but highly accurate. HDR predominates during S and G2 phases when sister chromatids are present. Thus, NHEJ prioritizes speed, while HDR prioritizes accuracy. CSIR NHEJ concepts. This slide summarizes highly important PYQ-based concepts. Site-specific recombination produces precise DNA rearrangements. Serine recombinases cleave all 4 DNA strands simultaneously. Tyrosine recombinases form Holliday junction intermediates. Lambda integrase requires accessory proteins like IHF. IHF functions as a DNA-bending protein. Excision stimulates excision but inhibits integration. In Cre-loxP system, orientation of LoxP sites determines recombination outcome. Cu70 and Cu80 recognize DSBs. Artemis processes DNA ends during NHEJ. HDR is mainly active during S and G2 phases. Finally, let us revise the most important concepts from this lecture. Site-specific recombination occurs at specific DNA sequences. Serine recombinases cleave all DNA strands simultaneously and rotate DNA subunits. Tyrosine recombinases form Holliday junction intermediates. Lambda integrase is a tyrosine recombinase. attB is bacterial attachment site while attP is phage attachment site. IHF bends DNA and assists recombination. Excision promotes prophage excision. Cre-loxP is widely used in genome engineering. NHEJ is error-prone while HDR is error-free. Cu70 and Cu80 recognize broken DNA ends. Artemis trims damaged DNA ends. RAD51 is essential for homologous recombination repair. Thank you. If you found this video helpful, please like, subscribe and share with your friends.